Hi everyone, my name is Dave AuCoin. I am out of the University of Nevada, Reno School of Medicine, and today I'm going to talk a little bit about some of the challenges my lab has faced developing rapid diagnostic tests for infectious diseases. Some of the data I'll show today involves the active melioidosis detect. It's a rapid diagnostic test for melioidosis. So here's a disclosure statement regarding that. So my laboratory works on a lot of different projects shown here, and our research focus is microbial biomarker discovery. So what is shed or secreted by a microbe during an infection that we can detect. We make a lot of antibodies. We do a lot of MAB analysis. We have a BSL-3, but importantly, we have a lot of the equipment to manufacture prototype lateral flow immunoassays or dipstick tests. We also have a startup on campus as well. That we use for submitting small business grants. But some of the research I'll go over today focuses on a bacteria called Burkholderia pseudomallei. It causes melioidosis. I'll talk a little bit about Francisella tularensis. This bacteria causes tularemia. And Ebola virus as well, which was a very tricky test to develop. I also work on a hepatitis B diagnostic test that I won't talk about today, but we're trying to detect the core-related antigen of the hepatitis B virus. When the virus reactivates, this core-related antigen can increase in concentration, and that may signal to a clinician that the hepatitis C virus is replicating at really high levels and the patient would need to be put on antiviral therapy. Here are just some examples of some prototypes that we've made for different diseases. Of course, they're sprayed down with antibody on the test line and the control line. So we work with a lot of monoclonal antibodies, and this machine here will spray down the antibody for capturing an antigen, for example. And then this machine here is a guillotine, and it chops these tests up into little strips. This machine here is a BioCore, and this can be helpful to look at the monoclonal antibody, kind of rank the affinity of the monoclonal antibody compared to other antibodies, so we can get a good idea that, okay, this antibody may be a good, for example, a good capture antibody, because it has the highest affinity to the target. So in case you didn't know, just quickly how these LFIs work, this is just a little side view. And what you can see here, shown here in pink, this is the conjugate pad, and that contains antibodies that are labeled with the gold nanoparticles. So they actually have this like reddish color. So if you have a sample and a bacterial target is within the sample, you put the sample on the sample pad, it flows down to the conjugate pad where it interacts with an antibody that is labeled with a gold bead. So those will bind and flow by capillary action down to the test line and get captured by the test line. So all these tiny little gold beads line up and you get a nice signal at the test line. So this is what you would see at the test line, the sandwich immunoassay. So here are just some factors you may want to consider when developing a rapid test. First of all, what is your target antigen? So what is the bacteria or fungus, for example, shedding or secreting in abundance that you can detect? Or what viral protein? And so this is oftentimes kind of the most trickiest part, I think, of assay development. There could be some known target antigens. If you look in the literature, maybe you can find like a good glycoprotein or something that's secreted. When you're looking at a virus, for example, maybe a new virus, some viruses just have like 10 genes, like Ebola, and are only making like 10 proteins. Others have more like 100 or so, so it could be a little bit more challenging. But oftentimes, glycoprotein or even nucleocapsid protein of viruses are a good choice because they can be conserved. But when you're dealing with a bacteria, oftentimes there could be up to 3,000 proteins that are produced by the bacteria. Which ones are shed, which ones are secreted. That's kind of the hard to figure out, the best antigen. So oftentimes you can use some genomics or bioinformatics to predict, are these proteins on the cell surface of the bacteria, or are they thought to be secreted? Another factor is, after you find out which target antigen you want to try to detect, you have to make some monoclonal antibodies. So how many do you need? If you're going to make a rapid test, 1 to 2 monoclonal antibodies that bind to the target may not be enough. 2 won't be enough, probably, just because you want to be able to select the best antibodies possible. So we'll make about 20 monoclonal antibodies to a specific target, and then we'll test all of them in the capture phase and detection phase to see which one gives us the best analytical sensitivity or limit of detection. So this involves gold labeling each of the monoclonals, or HRP labeling for ELISA. So you can run, sandwich ELISAs, or just run individual LFIs with different MABs in the capture and detection pair. Another factor is sample prep. What's the best sample type? Blood, urine, saliva? Where is your antigen accumulating? That's super important. What's the concentration of your target antigen in a patient sample? Is a lateral flow immunoassay or a dipstick test going to be able to detect low concentrations of this target antigen? Maybe you need something more sensitive. So a good way to figure this out is using a quantitative antigen capture ELISA, so you can figure out exactly the concentration of some of these antigens in patient samples. So here's one example of trying to pick a viral antigen to detect via lateral flow immunoassay. So our lab was developing a test for Ebola virus. This was in response to the 2014 West Africa epidemic. But most of the tests that were developed weren't very sensitive. There were 3 that were EUA approved, emergency use authorized. And all of them targeted the VP40, which is the matrix protein, which is a nice conserved abundant protein in Ebola virus. So it seemed like a good choice. However, the existing tests weren't very sensitive. The other option would have been like the nucleocapsid protein, or even the glycoprotein as well, even though maybe you can get some changes in the glycoprotein. However, what we did was we immunized with virus-like particles, and they don't contain any genomic materials, so it's completely non-infectious. And these contain VP40 and also the glycoprotein. So what we wound up isolating is 14 glycoprotein monoclonal antibodies and 18 VP40 monoclonal antibodies. So we went ahead and tested each of these glycoprotein-specific monoclonals and VP40-specific monoclonals in the lateral flow format. Shown here is a reader, so we could make an LFI, put it in the reader, and then we could get a number based on the relative intensity of the test line. So in this case, we're testing the GP-specific monoclonal antibodies, and so what we try to do is put each monoclonal antibody in the capture and then also in the detection phase, and just test different combinations to see which pairing is best. So this shows here that you actually need a lot of monoclonal antibodies to make a really good test. And it turns out this pair here had low background, 0 signal on the reader, and had nice test line intensity, right? So this was a good candidate for if we were trying to detect the glycoprotein. And then here's the VP40 specific testing. We went with this pair here that showed no background and high signal as well. And this is just again, just to remind people, test line antibody, capture the antigen, and then detection with the gold labeled particle. But this is where it got quite interesting, because we started testing these prototypes. We actually shipped them to the University of Manitoba in their BSL-4 laboratory, and they did live Ebola virus testing from virus grown in cell culture, the Makona strain. And so interestingly, the GP-specific test was the most sensitive. So shown here is just the different dilutions of the purified antibody that were tested on these LFIs. So we had a limit of detection from this GP test at about 1 × 10⁴, and you can almost consider this as like plaque-forming units or virus per mil. So pretty sensitive test, versus the VP40 specific test was not as sensitive. So that actually was only at about 1 times 10 to the 5th plaque forming units per mil. So not as sensitive, but still pretty good. And this was just a competitor's test that was actually EUA approved. And that one was the least sensitive, it appeared, of all the tests. So, the obvious choice was VP40, and others have made VP40 tests that were EUA approved. But it turns out that the GP-specific antibodies were more sensitive and yielded a test with a lower limit of detection, which was very interesting. Okay, next I want to shift to a bacteria, and this bacteria, Burkholderia pseudomallei, causes melioidosis. This bacteria lives in the soil. It's endemic to Southeast Asia and Northern Australia. Mortality rates are very high in these regions. Most people die of sepsis. The bacteria is resistant to antibiotics, commonly prescribed. There's no vaccine, and the endemic areas are definitely expanding. And so this has about probably like an 80% mortality rate if you're not on the proper antibiotics. If you are on the antibiotics that are recommended for this bacteria, it's about a 40% mortality rate in Southeast Asia and maybe a 12% mortality in Northern Australia. But the government actually considers this a biothreat. It does cause about 100,000 deaths per year in these endemic regions, so it's still quite a serious pathogen, causing a lot of morbidity and mortality. The big issue, it is just so hard to diagnose. So, a patient eventually makes their way into a larger hospital, like maybe within a city like in Bangkok or something like that. And blood is drawn, and then 5 to 7 days later, the blood culture results are taken. And by that point, a lot of these patients have passed away because they've probably already been experiencing symptoms for a couple of weeks prior to even getting into the hospital. So you really want to be able to diagnose this infection rapidly if clearly it's such a devastating bacteria. Another big issue is that the bacteria does not accumulate in patient samples to high levels. So believe it or not, there's 1 bacteria per mil in the blood. So blood culture bottles, usually they test about 10 mils of blood, and about half of them come back positive when a patient is positive for melioidosis. Urine, there's more bacteria in the urine, but only 21% of urine samples are positive. There's more in like a sputum sample or a pus sample, as one would imagine, but you really would love to be able to diagnose the infection from a blood or a urine sample. But this gets back to the fact that we're dealing with a bacteria that has a lot of proteins and carbohydrates that it produces. So which antigen are you going to try to detect? So my lab developed this technology called in vivo microbial antigen discovery, or INMAD. It's a diagnostic antigen discovery platform. And so what it entails is you take a sample, maybe from a patient or an animal model of infection. For example, if it's blood, you maybe filter the serum, isolate the serum, and filter it. So hopefully this sample contains shed or secreted bacterial antigens within the sample. So this is kind of like a diagnostic sample. If we take this sample then and immunize a mouse, this mouse will develop antibodies to the shed or secreted bacterial antigens, okay? And then after a while, you harvest the serum from this mouse that we call INMAD immune serum, and then you can probe a bacterial proteome array. So each bacterial protein is spotted in duplicate, and you can see positive spots showing up here from the serum. Or you can simply run a Western blot or a 2D Western blot to see against like the bacterial lysate to see which antigens are coming up positive. So the positive bands shown here should be antigens, bacterial antigens that are shed or secreted into this original sample. So you hopefully have an antigen that you would like to detect. So we took that INMAD immune serum from the Burkholderia pseudomallei INMAD study, and we probed a Burkholderia pseudomallei bacterial lysate. And you can see we're seeing a lot of positive bands that are showing up. This is the pre-bleed sample shown here in this first lane, and we're not getting any reactivity before the mice were vaccinated from the serum, so the pre-bleed serum. So a lot of different proteins are popping up. But interestingly, this high molecular weight antigen was what we were interested in, because we pondered that this could be the capsular polysaccharide of Burkholderia pseudomallei, which is a very abundant and conserved antigen on the surface of the bacteria. So it turns out when we proteinase K treated this lysate, shown here, we only get signal in the high molecular weight antigen, telling us it's probably a polysaccharide, and the proteins kind of go away. All right. And so this was very interesting to us. We did later confirm with purified capsular polysaccharide that it was this CPS, right? This deoxymanoheptopyranose polysaccharide. And so that was our target antigen for Burkholderia pseudomallei. We thought that would be a good antigen. The same thing happened with the Francisella tularensis study. This is another bacteria. And we identified the lipopolysaccharide shown here. These are just O antigen repeats of the lipopolysaccharide molecule. And then some other proteins popped up too. So this InMat actually worked with Francisella samples as well. So we were lucky enough to generate a capsular polysaccharide specific monoclonal antibody by just simply immunizing mice with heat-killed Burkholderia pseudomallei cells. And shown here is just using that monoclonal antibody and probing melioidosis patient urine samples. So you can see this high molecular weight antigen is showing up in 4 out of the 5 urine samples from very sick melioidosis patients. And here's a positive control just showing Burkholderia pseudomallei. And so the interesting thing too is that all of these samples were filtered prior to running them on the Western blot, telling us that we're actually not detecting bacteria, we're detecting a shed antigen. So one really challenging part of actually making a diagnostic test is potentially finding a private sector partner to help you out in manufacturing. So we were lucky enough to have a relationship with this company, InBios International in Seattle, and they started making more prototypes for us. We can make them in the lab, but they made something that potentially could be ready for sale or going through the FDA approval process. We sent a lot of these lateral flow strips to a collaborator in Australia, and we showed that we could detect the CPS within all different types of patient samples. So this was important, and it had a pretty good sensitivity of 200 picograms per mil. So that's actually pretty good for a lateral flow and immunoassay. A lot of LFIs have a limit of detection of about 1 nanogram to 5 nanograms per mil. So this slide actually answers the question, or starts to answer the question of what samples should you test. So for example, if you had one lateral flow immunoassay, but you had a serum sample and a urine sample from a melioidosis patient, which sample would you test? Well, it turns out we developed a quantitative antigen capture ELISA to quantify the CPS in serum samples from melioidosis patients and urine samples from melioidosis patients. Now, some of the samples had no CPS in them at all, which was interesting. But in general, the urine samples seem to have a lot more CPS, so the capsular polysaccharide, in the samples, as opposed to the serum samples. So some of them have 450 nanograms per mil of CPS in the urine, where the highest in the serum was roughly 6.7 nanograms per mil. Now, these samples did not come from the same patient, collected at the same time, so we were interested in kind of figuring out whether or not this was always true. So with a lot of help from collaborators in endemic regions in Southeast Asia and Australia, patient samples were collected at the same time, so urine samples and serum samples from the same patient, and we wanted to just compare the amount of CPS in a urine sample versus a serum sample. And in most all of the cases, there was more CPS in the urine sample than the serum sample. So we figured that that was probably the better sample to be testing. And so this was kind of a nice finding, simply because with a urine sample, you can actually test a higher volume of urine. You can also do things like concentrate the urine sample, or maybe do some enrichment for CPS within larger volumes of urine, and that would allow you to identify more positive melioidosis patients. Because a lot of the melioidosis patients, even the urine samples, had very low levels of CPS in them. So this was very helpful. Okay, I really want to focus on the fact that you probably need a lot of antibodies if you're developing a rapid test. Not just one or two, but a lot to test. So in this case, this is Francisella tularensis specific monoclonal antibodies that are binding to the lipopolysaccharide, and you can see the O antigen repeat that forms this kind of ladder pattern. And this is just a Western blot probing a Francisella tularensis lysate. And you can see the different antibodies, same concentration of antibodies showing different intensities. So that could give you an idea that, okay, maybe 1FT7 or 1FT10 are strong antibodies. But it helps you kind of down select which antibodies you should be testing. So this table here, we ran a checkerboard ELISA. So what we did here is we put each antibody in the capture phase and each antibody tested in the detection phase. And we tried to estimate the lowest concentration of the lipopolysaccharide we could detect in a spiked sample. And so as you can see, there's a lot of variation here, right? And so if you look at, for example, antibody 1FT4 in the capture, or right here, and antibody 1FT10 in the detection, we have a very high estimated limit of detection of 56. So if you chose these two antibodies just at random and tried to make a test, you would not have a very good test. But for example, if you chose another one like 1FT7 and 1FT8, for example, that would actually make a pretty good test, because that's at about 0.35 nanograms per mil. So very important to rank your antibodies, to test your antibodies in the capture phase and the detection phase, and also the same thing when you're testing your LFIs. Test them in the capture phase and the gold labeled or detection phase as well. So here's just kind of an optimized antigen capture ELISA using two antibodies that we thought worked well in the capture and detection phase. And you can see the limits of detection of about 180 picograms per mil in serum and about 130 picograms per mil in urine. So that's actually pretty sensitive, so a good test. But this slide just shows you the challenges of trying to detect the LPS in patient samples. We actually acquired some samples from a collaborator from the country of Turkey. There was extremely limited in volume. We brought these into the BSL-3 laboratory. We filtered them to remove any viable bacteria, and then we could actually do the ELISA at BSL-2. But as you can see here with all the patient samples, only, roughly half of them were positive, but they were very close to the limit of detection of the ELISA. One was very high, but others were very close to the limit of detection of the ELISA, telling us that there does seem to be LPS in these patient samples, but it is quite low. So what we're working on now is maybe we can detect more LPS in a urine sample from a patient, but as you know, these samples are quite hard to acquire. So I went through that research pretty quick, but the bottom line is it took a lot of help from a lot of researchers within my laboratory, and then a lot of collaborators from around the globe, and funding agencies as well, like NIAID, Homeland Security, and the Defense Threat Reduction Agency.